and some people also from Indonesia. Hi, good afternoon. And some people also from Japan. Good evening. And in here, I also saw other people from other country because at least for today, we have participants from 8 different countries. So that's really wonderful. So on behalf of the organizing committee, I would like to welcoming you, a researcher, a student, and also the professional from across the country for today's international webinar. My name is Yolanda Antonius, and I will serve you as your master of ceremony and moderator in this seminar. And for the agenda, we would have 4 amazing speakers today who will share about wonderful topic in biotechnology. But just quick reminder, we will have 5 exciting door prize as a voucher for the proofreading and also publication assistant service. So for anyone who want to join, just take a picture during the event, share in Instagram, and don't forget to tag our account. Then our team will pick the winner from your post. So for officially open the webinar, I'm happy for welcoming Dr. Arif Ansori. He is a founder of the Virtual Research Center for Bioinformatics and Biotechnology, VRCBB, for delivering the opening remark. So, Dr. Arif, time is yours.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone, and warm greetings to everyone that coming here to this event. It's my great honor and pleasure to welcome you, all of you, to our international webinar series, jointly organized by the Virtual Research Center for Bioinformatics and Biotechnology Indonesia in collaboration with Jalan Tengah. On behalf of the committee, I would like to express my heartfelt gratitude for your presence today, especially to our distinguished speakers, collaborators, and their enthusiastic participants from Indonesia, India, of course, and other countries. And special thanks to all of my colleagues, Dr. Mada Triandala Sibero, a senior researcher of VRCBB, and Ms. Shrabonti Chatterjee from Visva-Bharati University, and Dr. Kavitha Kandiah from Rangasamy College of Technology, India, Associate Professor, and also visiting researcher in National Research and Innovation Agency, or BRIN. And last, Dr. Muhammad Fahmi, senior researcher from Anagenic Indonesia. This webinar marks an important milestone in strengthening international collaboration between particularly Indonesia and India in the fields of bioinformatics and biotechnology. These two rapidly evolving disciplines are at the forefront of innovations that can shape the future of healthcare, agriculture, environmental conservation, and many more. I would also like to extend my special thanks to our technical and organizing team for making this event possible, and to our supporters, snplans.com and fundsbiotech.id. Please enjoy the webinar and make new connections among us, and take this opportunity to explore potential research collaboration and knowledge exchange. Once again, thank you and welcome. Wassalamualaikum warahmatullahi wabarakatuh. Terima kasih and dhanyawad.
Okay, thank you so much for Dr. Arif for the word. And now we are moving to the first session, but before that, I will share the CV for our first speaker. It's an honor for me to welcoming Dr. Mada Triandala Sibero. Hi, Dr. Mada. Are you there?
Hi, can you hear my voice?
Yeah, it's clear. How are
Oh,
you?
wonderful. Feeling great, but
Oh,
pretty cold
yeah.
in New Zealand today.
I see. What time in New Zealand today?
It's 6:20 p.m.
Oh, it's already night.
Yeah, like
I
five
see.
hours different.
I see, I see. Okay, before we start for the presentation from the Dr. Mada, so let me introduce him just for a while. In here, he has concern in microbiology and bioactive compound. And as you mentioned before, now he stay in New Zealand as a mycologist. And he also had many, many experience, for example, as the postdoctoral researcher in China, also the visiting scientist also in China but in different university, and also he was a lecturer in Indonesia. And regarding for the research, he has so many publication and research that already published in international conference. So we don't doubt that he will share something that really interesting for us. So for Dr. Mada Sibero, so time is yours.
Thank you so much, Miss Yolanda. Hi, everyone. Good evening from New Zealand. Well, for today, I will share more or less related to my expertises, which is microbiology. Wait, I'm trying my best to. Yeah, this one. Wait a minute. Yep. Can you see my slide?
Yeah, it's clear.
Okay. Wonderful. So, hi guys. Again, my name is Mada. You can call me Mada, and currently I'm living in New Zealand, and I'm a full-time researcher at VRCBB together with Dr. Arif. And for today, I would like to present my previous work related to exploration of novel bacterial species from mangrove ecosystems, and later I will also explain a little bit about how to obtain their secondary metabolites, especially if you're looking for new compounds. Yeah. So, the reason why I'm really interested on microorganisms because as simple as they are a lot in our environment, including in our body. So this is one of the picture that's taken by other scientists, which mentioned that in 1 liter of seawater, it contains more than 1 billion prokaryote cells and virus. And then we can say that microorganisms has plenty of roles and function, yeah, including in biogeochemical cycling. Therefore, we can use all the nutrients in the environment. Then they also have a very important roles for ecosystem restoration and bioremediations. And then they also has a very important roles to maintain our health because some of them they also causing diseases. And the last but not the least is related to the bioprospect. So as you know that plenty of antibiotics nowadays or even the commercial enzymes, most of them are produced by microorganisms. And these all microbial roles and functions impacting the human welfare. Therefore, microorganisms are very important. We cannot forget them. We cannot neglect them because once we neglect them, COVID-19, one of the problems will appear in the future again because we forgot or we neglect the presence of these microorganisms. And if you're talking about microbial bioprospecting, especially bacteria, actually, sorry. So bacteria actually has been explored for many, many years. So, for example, we are concerned on on antibiotics. So, if we check all the list of antibiotics until today, most of them are produced by by bacteria. Well, there are several exclusion, which is penicillin. It was the first natural antibiotics produced by fungi. But the rest of them, most of them are produced or what do we say? It is inspired, yeah, for the chemical synthesis, but still the source is the natural compounds from bacteria. And most of them, yeah, for the list of antibiotics that I gave in this presentation, well, they are produced by one genus, namely Streptomyces. Although Some other antibiotics, they're produced by other genus. For example, in this picture, you can see Micromonospora, Scaropolispora, or Bacillus. And do you know the reason why most of the antibiotics nowadays are produced by this genus Streptomyces? Because Streptomyces, I can say that because, well, this bacteria is part of Actinomycetota, or previously known as Actinobacteria, or Actinomycotota, or yeah, all the terminology. But Streptomyces is the easiest Actinobacteria that you can isolate, as long as you give the media enough soluble starch, and then you give the media antifungi, and if you have nalidixic acid, you can add it because nalidixic acid is another antibiotic to inhibit the gram-negative bacteria, and you let your agar plates in isolation in the um, what do we say, the incubator, yeah, you can isolate plenty of streptomyces. Why? Because streptomyces, it produce heaps of aerial spores, and they are really, really fast growing. I mean, like within 7 days, you already can see the spores. Or, for example, if you are working with enzymes, I do believe most of the papers that you read, they were produced by Bacillus. Why? Well, the same reasons, because Bacillus also one of the easiest and fast growing bacteria that you can isolate. So no wonder if you read papers, most of them will produce by Streptomyces or Bacillus. If you're working with fungi, maybe most of your reference, they are working with Fusarium, Aspergillus, Penicillium. Well, the reasons are the same because they are very easy to be isolated. So the case now is the antibiotic resistance is increased. Yeah. The reason why, because, well, plenty of reasons, including the irresponsible use of antibiotics. Another reason is because sometimes you don't really dispose your antibiotics shells in a good ways. You just throw it anywhere, so it can also increase the antibiotic resistance in environments. Therefore, the investigation of novel antibiotics also increased, but the new novel, or sorry, the discovery of novel antibiotics is decreased because, yeah, most of the scientists, they re-isolate the same antibiotics because they also isolate the same bacteria, which is Streptomyces and so on. On the other hand, yeah, in fact, less than 2% of bacteria are culturable and well identified. So it means that we have eight, at least eight, the 98% of bacteria species that are neglected or underexplored. Therefore, to isolate new compounds, you can use this strategy, which is explore rare genera. So in my work when I was in China, so this presentation is the data that I obtained when I was working in China as a postdoc. So our lab focused on exploration of actinobacteria. But later, during the isolations, I also isolated some others genera which not from actinobacteria. I will explain later. But the point is, we try to obtain new antibiotics from rare genera. And why we cannot, why we should isolate the rare genera or the species that never been isolated before? Because how do we can work with something that we don't know their capability, or even we don't know who they are? Just like bacteria, you cannot utilize certain bacteria if you don't isolate them. And this is the biggest challenge during the explorations, either for novel species or novel compounds or novel medicines from microorganisms. The biggest challenge is when do the isolation. Because if you do a isolation and then you just do and repeat the same protocols, the result is you will isolate the same bacteria again, again, and again. And in this work, I focus working with mangrove ecosystems. Why? The first reason, because mangrove ecosystem, they are exist in tidal area. So this tidal area will give them a fluctuative salinity, pH, temperature, moisture, and so on, which also will give a very strong correlation with the microbial diversity within. And mangrove ecosystem, especially in Indonesia, also very high pollutions, and somehow the high pollution will also give them stress that will induce several of their genes to activate and produce secondary metabolites to protect themselves. And then, of course, the UV exposures will also give them stress and will induce their ability to produce secondary metabolites, high nutrient, and also host microbial interactions. Because mangrove itself, they have their own holobionts, and their holobionts also very interesting, but kind of neglected, I guess. Therefore, in this research, we focus to work with the mangrove ecosystem microorganisms, especially actinobacteria. And the last reason why I work with mangrove ecosystem, well, because I'm an Indonesian, and in my country, Indonesia, yeah, we have plenty of mangroves. They are so diverse. They have plenty of um, species, but in the location that I work with, which is Semarang City, most of them are covered by Rhizopora apiculata or Rhizopora mucronata, and another side uh, occupied by Avicenna marina. And then if you're talking about the results, yeah, again, like what I've told you about the isolation, so during the isolation and purification, my colleagues always mention to me that, well, Mada, actinobacteria mostly they produce aerial hyphae. Yeah, so they look like fungi. So just try to isolate all of the aerial happy colonies. So I did what they suggest, but then I got that most of the actinobacteria or most of the strain that I isolate are belongs to Streptomyces. And in that time, the diversity was very low because I only got Streptomyces, Amylocotopsis. I also got Micromonospora, only those genera with some others minors, hyphae producing or mycelium producing bacteria. And then I started to think, oh, how if I also isolate others colonies which not produce hyphae? Maybe they are interesting. My first trial, I worked with these big colonies. You can see that it's like very beautiful color, pink. But when I did 16S RNA gene sequence analysis, well, it turns to Bacillus. So Bacillus, it's not actinobacteria. So I lost my interest. Then I isolate another colonies, which did not produce mycelium. It exactly looks like common bacteria. Then I found that this bacteria is Nocardioides, which is an actinobacteria. So I found that actinobacteria is not always produce mycelium or hyphae or like fungi colony like, but they also might produce like a general colony just like a bacteria, but well, they are actinobacteria. So I keep continuing isolate any bacteria that grow on my plates. But you can see on the pictures, in the left pictures, my plates were not really were not really much of colonies. The reason why, because I use 9 different media. I use different treatments. For example, like heat treatments. I also add some chemicals. So those heat treatment chemicals, UV treatment will also decrease the number of untargeted microorganisms. Yeah. And then I also, of course, I did some antibiotics, which is cyclohexamide, that's to kill the fungi, and nalidic acid to kill the gram-negative bacteria. However, when I did genetic work and I did the 16S RNA gene sequencing, and then I did the analysis, I found that my strains, they were not only actinobacteria, or nowadays they rename it into actinomycetota. Yeah, but I also found several Bacillotota and Pseudomonadota. So you can see here that I got like 300 actinobacteria and then 137 Firmicutes previously, namely Firmicutes, and then I got 47 gram-negative bacteria. And although I isolated more than 600 strains, but only 484 isolates were well grown and successfully identified. Why? Mostly the reason because they are dependent on other strain. What does it mean, Mara? So, for example, in these pictures, you can see that the green. Okay, let me use my yeah. Okay, for example, this is the colonies of my bacteria, right? So it produces a very beautiful green spores. Then I tried to take under microscope and I found these white colonies. So I thought, okay, these white colonies, maybe it's also part of this bacteria. So I just directly sequenced them, and it turns that these colonies is not single colonies. So I used another 12 media to purify them, but they cannot be purified. They always grow together. Therefore, when I try to identify them, I cannot. So they are simply unseparated. Therefore, I only get 484 isolates that I can identify. So this is the result for the bacterial composition. For the first is Actinomycetota or Actinobacteria. So you can see here, I successfully isolated 33 genera, and several of them are rare genera. What does it mean, rare genera? So rare genera, you can say it is rare genera if number one, they are like reported or less reported or barely reported. Number two, because the species, yeah, the number of species from this genera is less than 10 species. For examples, Tersicoccus, yeah, you can check my paper. I just published one new paper related to the new species from Tersicoccus. So Tersicoccus, this actinobacteria only have two species. Yeah, first is Tersicoccus soli, and another one Tersicoccus phoenix, if I'm not mistaken. So I reported the third species from this genus, Tersicoccus mangrovi. So that's how we say this one is rare genera, this one and this one is not rare genera. And then from actinomycetota composition, yeah, I also do of a Venn diagram analysis, and I found that from six different locations, yeah, Mangkang, Tirang, and Meracocho, I do two sites for each location. So from six of them, I found Streptomyces and Rhodococcus in six locations. So it means that Streptomyces and Rhodococcus, they are everywhere. And the same result also I found when I compare based on the isolation media, where Streptomyces and Rhodococcus, they can grow in all media. Therefore, I mentioned to you the reason why people always report about streptomyces, because simply they are very easy to be isolated. On the other hand, some rare genus, for example, like Tarsicoccus, Actinoalomurus, or Mumia, Esotericola, they cannot grow in like general media. You need to uh like understand their physiology, and then you can put your own recipe and try to isolate them. Yeah, so isolation and purification is very tricky, because if you are lucky, you will get like very rare genus. But if you are in the like very for example, like you are in rushing time, you will repeat the same protocols and you will get the same genera. And for the Brasileota compositions, although I only obtained around 137 isolates, yeah, but this is pretty good data, because I found that they are from 20 different genera, and some of them are very rare. For example, is Simazuela and Corylibacterium. And for the gram-negative bacteria, yeah, again, why the number of isolates is very low? Because I added nalidixic acid, which killed the gram-negative bacteria. Therefore, I only get 47 strains, but from 47, I got 15 genera, also pretty good. And I will explain later about Frigidibacter, because I found new species from this genus. And Giela, I also found new species from this genus. Then, Okay, Mada, after we obtain hundreds of bacteria and we want to continue for the biotechnology or bioprospecting, what we should do? If you're focused or concerned to obtain bioactive compounds or new compounds, I can suggest you two common ways that I have done before in my previous work. First approach is chemical screening. So for chemical screening, you can do what I did when I was studying in Japan. So I use HPLC DAD or diode array detector. So using HPLC DAD because you will find the UV pattern. For example, you already got 300 bacteria, so you need to culture all of them in liquid culture or agar culture, no matter which one. Sorry, and then you do extractions and all the crude extract, the 300 crude extract, you inject to the HPLC. So for example, if you use the HPLC DAD, yeah, you will get this chromatogram, yeah, and then if you click the chromatogram, then the UV absorption pattern will be appear. So chemical screening, you can do it if you have a library or database. So in my previous home base, Toyama Prefectural University, under Professor Yasuhiro Igarashi, our lab, we have UV library contains more than 2,000 compounds. Therefore, every time we click the pick and then we try to match with the library, okay, we can know, okay, this pick, yeah, has this UV pattern, and this UV pattern has similarity, yeah, with divergolide. So once we know, okay, this pick is not a new compound, we will not continue to work with that. We just keep, and then we will keep continue to find the pick with a unique UV spectrum that never been recorded in our lab. So for the pro, yeah, this chemical screening almost guaranteed to isolate new compounds. Yeah, well, 90 to 99% because Igarashi lab, they reported plenty of new compounds using this chemical screening method. But the constraint is it doesn't guarantee the biological activity. Therefore, we isolate plenty of new compounds and then we don't really care about the biological potential. For example, like is it like potential as anti-cancer or anti-inflammation or antivirus? We don't really care. As long as it is a new compound, then we will keep continue to work with that. For example, this is my previous work when I did my postdoc. Yeah. So I cannot disclose because this data is still under review. Yeah. So in here, I work with one of my coral associated bacteria, not from mangrove anyway. Then in here, you can see this is the UV chromatogram that I obtained from one of the fruit extract. And then when I click in this is the UV pattern that I got. And this UV pattern never been reported in the exact detection time. Then we try to obtain this UV, this peak, sorry. We try to obtain this peak. We try to collect them. Therefore, I did the open silica chromatography to do the fractionation. So I did 2 times fractionation using silica and using ODS, autodiesel silica. And then every fractionation step, yeah, I will inject all fractions to the HPLC, and then I will looking for this peak. And then I found in the last fractionation, in fraction 40% of acetonitrile, I found this peak. And later, after I do isolation, and then my professor helped me to do the NMR and all the elucidation structure, we confirm this compound as a new compound or novel compound. But this compound has no antibacterial activity because in our lab, we did antibacterial screening, but we are not really sure whether these compounds are potential for other biological activity. Therefore, this kind of collaboration is very good, yeah, between someone who working with isolation and someone with who working with in silico, like Dr. Arif or Mas Fiol. So all the new compounds you can do the screening through the in silico, and if it's potential, you can continue for the in vitro or um, in vivo assay. But because now, well, this data I obtained, and then until today, we try to figure out what kind of activity that it has. Okay. Second thing is based on bioactivity screening. So based on bioactivity screening, I did this in my postdoc in China. First, for the pros, it guaranteed the biological activity. What does it mean? So it means if you are looking for antibacterial compounds, so you will do bioassay for or antibacteria test in every step. So therefore, the compounds that you will get, it's supposed to have antibacterial activity. But the cons, you will re-isolate plenty of known compounds. So barely can isolate new compounds as a new antibiotics. Therefore, people who working with bioactivity screening base, they will spend plenty of times because if you want to publish a paper like a good paper in Journal of Organic Chemistry, you need to publish a new compounds with bioactivity potential like antibacterial, anti-cancer, and so on. Therefore, bioactivity screening is pretty interesting for someone who working with with the biological potential base, but it will take a long time. Well, my previous place in China, our lab focused on antibiotic discovery. We have several PhD students who were in their fourth year, but they are still trying to obtain new compounds because they are focusing on new compounds with antibiotic properties. Yeah. Therefore, another things that we develop in our lab in China, yeah, after we obtain, okay, this species or this strain, it has antibacterial activity, right? So we want to confirm, is that bioactive new compounds or not? Sorry, is that metabolite new compounds or not? Well, before we do like big production, spend plenty of hours in the bench, we will do genomic based like analysis. So simply we will do the whole genome sequence, and and then we put the whole genome sequence to anti-smash, and then we will check for the PKS, yeah, polyketate synthase, and NRPS, non-ribosomal peptide synthase. If these genes, yeah, exist, and then we will check the similarity. If the similarity is very high, like 100%, we will skip this strain. But if this strain has like low similarity, for example, the beta lactone is one of the group of uh, compounds that also has good antibacterial activity, and the similarity is very low, and there's no other reports from this species about this genus, Nocardioides, about for the antibacteria. So we continue to work this with this strain. Therefore, some other students, yeah, currently they are continuing for the exploration of antibacterial compound from my strain. Okay, that's all about the exploration of novel compounds. Now I want to give you a view knowledge, I guess, related to polyphasic taxonomy. So what is polyphasic taxonomy? Basically, it is the combination of genotyping, phenotyping, and biochemical characterization to identify and confirm a new species. Sorry. So for this work, this work is very simple actually, but very tedious. But in Indonesia, no one really, I'm not really sure. Yeah, in Indonesia, only a few people. Yeah, only a few people do these works. I know some experts in BRIN and in University of Indonesia working with identification of novel species. However, report from Indonesia is pretty scarce. Therefore, for anyone in here who are interested working with new species, you can contact me, and I will do my best to help you to identify your species or yeah to guide you to to work with novel species from Indonesia because our country is so rich, but only few people doing this work. Well, let's continue. For the genotyping, you can work with the whole genome, and through the whole genome, we need to calculate the average nucleotide identity, our ANI value, and then you need to do the genome and genome annotation and comparison. Then you need to get the data for digital DNA hybridization value, and the last but not the least is phylogenetic analysis. And then for the phenotypic, you will work with plenty of plates and plenty of liquid media to determine which media is the optimum to grow them. And then you will also do some works, yeah, very tedious, to determine their optimum pH, optimum temperature, and optimum salinity to grow your new species. The last but not the least is biochemical analysis, including cellular fatty acid, respiratory quinone, polar lipid, the amino acid, sugar consumption, hydrolysis activity, and enzymes activity. So it's not difficult but very tedious. So let's start with my previous work. Yeah, for the screening of novel species. For example, guys, you are working with 16S rRNA gene sequence. So mostly, I'm not really sure in other countries, but according to my previous experience working in Indonesia from 2017 until 2020, yes, until 2021, most of students that I met, they only work with 100 until 1000 until 1300 base per sequence. Is it good or bad, Mada? I can't say it's bad, but actually it's not enough to determine a species. Because, yeah, for example, although the similarity is 99%, it might be a new species. Therefore, usually in our lab, we will use these three characteristics to choose which strain we should continue. Number one is the similarity. And then, for example, in this picture, in this table, you can see my strain number 98. It has similarity 97 and 97 point something percent with Gela endophytica and Gela mangrove. Okay, usually if you have similarity less than 98%, yeah, remember less than 98%, it might be a new species. Okay, and then after we found, okay, the closest species are Gela endophytica and Gela mangrove. One thing you need also to remember, if you want to work with novel species, you also need to make sure you have the reference strains. So what is reference strains? So reference strains is the strains that has lots of, yeah, similarity with your strain, yeah, that you will use to compare. What things that we need to compare, Mada? This. You will compare your strain with the reference strains based on their genome, based on their phenotype, and based on their biochemical activities. So therefore, you need to check, are the reference strain available in your place or not? Okay, and then you also need to consider the number of species from the genus. For example, if you work with streptomyces, well, streptomyces has plenty of species. So if you have plenty of species, it means you need to compare your candidate of novel species with more reference strains. You cannot use only two or three if you work with streptomyces. You need to compare them with at least four reference strains. Therefore, in our lab, we never work with streptomyces, although we usually isolate plenty of novel candidates, yeah, novel species candidates from streptomyces. But we are very interested to work with bacteria or from rare genera bacteria. So, for example, I will continue to explain about strain code number 98. Yeah. So, for example, after you decide, oh, okay, this one is the candidate of your new species, you need to determine their ANI value and DDH value. This value is very, very easy, yeah, because everything is already in platform, like online platform. You just upload your data, and after you upload your data, the system will calculate it for yourself. So you don't need to to wait a long time, only like 6 hours, yeah, and then you don't need to do anything else, and then you directly get okay your NI value within the closest or within the reference strains is 77%, and then your DDH value is around 20%. So guys, according to the standard, if the similarity for an ANI value within your strain and the reference strains less than 95%, and the DDH value within your strain and the cluster species or the reference strains less than 70%, you can confirm, okay, this one is a novel species or new species. Then, yeah, for example, code 98, you can see here the NI value, sorry, the NI value less than 95% and the DDH value less than 70%. Then I can confirm, okay, strain code 98 is a new species or novel species. After that, after you get a confirmation from ANI and DDH value, then you can continue with the phylogenetic analysis. So usually for the new species, you need to provide two phylogenetic trees. Number one is based on the almost complete 16 sRNA gene sequence. So you cannot use only 1,300. At least you need to provide 1,450 base pair. Okay. And then after that, you can check here. Oh, sorry, where's my, okay. You can check here the bootstrap number is very low, 66. So it means within genus Gela, the position of my new species is not stable. Therefore, it might be a new species. And then when I continue to build a phylogenetic tree based on the core genes, yeah, within the whole genome, you can see here my new species. It forms a new clade and separates it with other Giela. Therefore, we can suggest, okay, these trends might this trend is a new species. So anyway, because currently the paper is under review, we name it as Giela semarangensis. And then again, just like what I explained before, you need to do those tedious work to determine the media, the physiology for salinity, pH, and temperature, and then you also to understand their ability to hydrolyze several macromolecules such as starch, cellulose, milk, gelatin, citrate, twin 20 until twin 80, and then you can also use API kit. Yeah, API kit is very common. Yeah, to understand their ability to ferment. Yeah, to do fermentation using different sugars and also to check their enzymes activity. And for the another thing that's very important is the polar lipid. Yeah. So for the polar lipid profile, yeah, you need to extract the polar lipid, yeah, using hexane, and after that you can do the two-dimensional TLC to determine which lipid is sorry, which spot is belong to certain lipids. For example, in here, for Giela, yeah, for Giela, uh PC is phosphatidylcholine. This is the specific, not specific. This is the special polar lipid that contains in all Gela species. Therefore, we can confirm, okay, this species really belongs to Gela, and this pattern is very important to explain to the readers and to convince the reviewer that you do a correct way to determine a new species. And another one is the amino acids. The amino acids usually for GLA, yeah, we obtain well for the amino acid we only use simple TLC, yeah, and this the amino acid is very important to check the presence of special amino acid in your new species. And in my new species, I confirm there is the amino acid in my species is meso deaminophilic acid or DAP. And the other one is fatty acid. So basically, for fatty acid, we only check the difference between our strain and our reference strains. Is there any difference related to the what do we say? Yeah, uh, the abundance of the fatty acids and also the diversity of the fatty acids. For example, in my strain, there is no C18. Yeah, there is no these two unsaturated fatty acids. So we can say, okay, for my strains, this is the specific condition for the fatty acid content in their cells. And the last but not the least is related to the certificate. So if you guys want to publish a new species or novel species, you need to get certificate from culture collection, at least from two different countries. So for this new species, I registered them in Korea and also in China. I think that's okay. I still have several minutes, yeah, I guess. So if you want to do the bioprospecting again in your in your strains, you also can do the genome analysis, yeah, but I'm not really sure is it like cheap or not, but in China it was pretty cheap, therefore we just send all the candidate strains and then we do anti-smear analysis, and you can compare here between these novel species from Nocarioides. It has antibacterial activity, yeah, and it only has a VU polypeptide synthase and well BGC or biosynthetic gene cluster. However, when I compare to another Actinobacteria novel species from genus Actinoalomorus, you can check here the biosynthetic gene cluster were very diverse. Therefore, the potential of this species to produce new compounds is bigger. Therefore, another PhD students also continue these strains. And why this strain is very interesting, yeah, you can see here these strains can inhibit itself. So this is the young colonies, and these are the old colonies. And somehow the old colonies produce antibacterial compounds to inhibit the young colonies. Therefore, we continue to work with this special strain. And Mada, why do scientists prefer to work with actinobacteria rather than others? Well, simply because the biosynthetic gene cluster in actinobacteria are more diverse. You can see here in Simazuela, it is not this one is not an actinobacteria, but belongs to Bacillota. The BGC is very scarce, very, very low in here, including in Gela. Most of them are just pigment producing genes, including the Frigidibacter. These are two novel species that I am writing right now. Yeah, this one even it don't have any. It doesn't has any biosynthetic gene cluster. Therefore, people usually work with actinobacteria to do like exploration for novel or bioactive compounds. I think that's the end of my presentations. These are the teams that I work with of Professor Cheng Hasun from Institute of Medicinal Biotechnology in Beijing and Professor Tuoli from Chunyi Medical University. Thank you very much and have a wonderful day.
Thank you very much, Dr. Mada. That's a really wonderful talk. And it's really exciting for having a research about the exploration for microbiota. That's really interesting. Now we are heading for the Q&A session. So if anyone had a question, so please do raise your hand or you can write your question in the chat box. And in here, let me check. In the chat box, we have two questions so far. And in here, we have Mr. Fiol here. He from University of Airlangga, Indonesia. He asks about the superbug treatment with the prospect of the bacterial species from mangrove. Has it been done before, Dr. Mada?
Okay, for the superbug treatment, like
Hmm.
for example, methicillin-resistant Staphylococcus aureus, yeah, or extended bacterium, extended spectrum beta lactamase, any gram negative. I think plenty of works they already try to isolate compounds from mangrove associated bacteria. However, I'm not really sure is there any new compounds that has ability to inhibit or to kill the superbugs. Well, you can check my paper related to TMKS8A. That is one of the new compounds that we isolated from mangrove associated bacteria. Well, it is a new compound, but it has no antibacterial activity against any superbugs. So this is also very interesting to be done. So if anyone who want to work with this topic, it's very good to be explored. But remember, if you want to work with superbugs, you need to work with them in the biggest even start from the beginning. I mean, like most of scientists in that I know, they will work with ATCT strain. For example, okay, we want to find the bacteria to inhibit multi-drug resistant bacteria, but their work pipeline, they use ATCC or they use the non-clinical type or they use the non-MDR type strain. So when they do the work and then after they get the compounds and then they combat with the drug resistance or with the superbug, that compounds is useless. Therefore, I suggest if anyone who want to work with superbugs, use them start from the beginning. So you will get your target compounds. Yeah,
Yeah, it's look like a screening, right? So
yeah.
we
For
should
the
start from the
screening,
beginning.
yeah, start from the screening. Just use the superbug. Do not use the sensitive pathogens. That's the point. Yeah.
Thank you so much. So hopefully that can answer the question from Mr. Phil. So we are going to the second question. It's from Mr. Ahmad Zainur. And in here, he asks about the do biosynthetic gene cluster also applicable on other organism for plants, for example?
Yeah. Hi, Ahmad. So for the biosynthetic gene cluster, it also exists in most of the organisms, including plants. Even if you are looking for anti-smash, if I'm not mistaken, it also has version for plants. So yeah, they are exist in plants, and you can explore a lot of
Okay.
biosynthetic gene cluster in plants, and you can. Well, some of my colleagues in United States they work with what do we say the gene modification in plants, how to improve the production of certain compounds. So they just knock out the BGC in the plants. Yeah, so yeah, this kind of work is very possible.
Okay. Thank you so much, Dr. Mada. Now we have a new question. This is from Mr. Joydeep Mahata.
Mhm.
He had questions. So, hello, Mr. Joydeep.
Hi.
Are you gonna talk directly or just me that reading your question?
Hello, am I
Hello.
audible?
Hi.
Hi, a wonderful talk, Mada. I
Oh,
had some
thank you so
questions
much.
that you,
Hmm.
and at the very last, you said the bacteria colony, when it is getting old,
Hmm.
it is emitting some substance itself that is inhibiting its own population, younger population. Is it affecting its own species, or is it can affect others bacterial species as well?
Yeah. So according to our further test, it also affects another species, especially for the drug resistant Pseudomonas aeruginosa. So, but it's only affects in certain species like Pseudomonas, and if I'm not mistaken, another one is Klebsiella. Yes.
Okay.
So, but we are not really sure. Is it like the same compounds that inhibit themselves and the pathogens, or they produce different? Because we do screening when they based on like antagonistic tests, not the extract with living cells. Therefore, the PhD students still working with that. Hopefully, we will get the data and publish a paper within next year, I guess.
Oh, nice.
Yeah.
Thank you, Miss Mr. Joydeep. Okay, now we are going to the next question. This is from Mr. Suhartomi from Faculty
Mhm.
of Medicine, Universitas Prima Indonesia. Hello, Mr. Suhartomi. If you want to like discuss directly, it's also possible. Or maybe I also can read your question. Hello, Mr. Suhartomi. Okay, I guess I will read the question. So in here, he asks about the application or the purpose
Mhm.
for isolating the new species, because is the focus on the for utilizing the metabolites, or it's you aim to develop a new life probiotic product?
Oh,
Yeah,
very
that is,
wonderful question.
yes.
Yeah. So our lab in China, you can check at Institute of Medicinal Biotechnology, Chinese Academy of Medical Science. So in that lab, our focus is to utilize the secondary metabolites. So we try to isolate as much as possible rare genera and then isolate the novel compounds, and then we will try to isolate their secondary metabolites. But in another what do we say, kind of like purpose, of course, someone can use our strains to develop another products, including probiotics. But again, I'm not really sure for probiotics because usually, especially in New Zealand, for example, I'm talking about New Zealand. In New Zealand, you cannot use like wild type to make to produce probiotic. You need to like have a GRAS certificate. Generally, sorry, I forgot the G, but relatively safe, if I'm not mistaken. I forgot the G. Well, the point is you need to make sure that your strain is safe and will not produce toxins and will not make any like problems in the future. Therefore, like including in China as well, we cannot develop that kind of probiotic that easy. Therefore, most of the laboratory and people that I met in our institute, we work with the secondary metabolites. Hopefully, it answered your question.
Thank you so much. So basically, everything that we collect is it should, yeah, we just aim that hopefully they have like some function that we could apply for the human, right?
Yeah, correct. Another thing that we work with another lab that we have collaboration is their enzymes actually. So
Hmm.
some of the can I share the pictures? Well, please
Sure.
wait a moment. I think uh, mm, 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 mm. because w- my lab also collaborate with other. Okay, this one colony. No, unfortunately, I forgot. So sorry, but we work with another lab to work with arginase and also
Hmm.
tyrosinase, and those enzymes are currently developed to make medical enzymes because if I'm not mistaken, it is very beneficial to treat cancer. Yeah, but it is done by other lab. I just provide and do
Hmm.
the screening, and then I provide the strains to other lab. Yeah.
Okay, thank you, Dr. Mada. And the
Okay.
last question, this is
Mhm.
from Mr. Zahid Salim. Hello, Mr. Zahid Salim.
Uh,
Are you going to have a discussion directly? Okay, maybe I will just read his question. he asks about the specific challenge that you face while you isolating the novel of bacteria, especially from mangrove ecosystem. Do you have any like challenge when you do that?
Oh yeah, I think the biggest challenge is during the purifications because again, plenty of bacteria they are dependent to each other. So for example, the picture that I showed before that I cannot separate them because if I separate them, they won't grow. Or when for example, they grow in our isolation media. Then when we purify, they still grow, but the second purification or when we reculture them, they won't grow anymore. The reason why, maybe because the temperature change, the
Mhm.
salinity change, the humidity change. So it's kind of the things that are very challenging. But once you get a stable living candidates, it's very easy to continue. You know what? I can finish to determine new species, like 5 new species within 2 or 3 months. So this
Mhm.
one is a very, very easy work, but very tedious. That's all.
Mhm. Yeah, yeah. As you mentioned before in the slide, there is like some colony that can be separated, right? I guess
Yeah,
that's
yeah.
one of the challenges that you should face. Okay, thank you so much, Dr. Mada Triandala Sibero, for your insightful talk. It's really, really exciting to know about the exploration of microbiota. Thank you so much. And
My
now we are
pleasure. Thank you
going.
so much
Bye
for having
bye.
me.
Thank you so much. Yes. Thank you so much. So now, after we talk about the bacteria, now we are going to the second speaker, and we honor to welcome Miss Shrabonti Chatterjee. She is a PhD student at Visva-Bharati University, India, and also a member of VRCBB Indonesia. Hello, Miss Shrabonti.
Hello. Yeah,
Hi, how are you?
I'm fine. How are you?
I'm
So
good.
am
I'm
I
really
visible?
exciting to hear about your presentation.
Yeah, sure.
Okay, so but before we are going to hear the presentation from Miss Rabonti, so let me share
Okay.
for a while about the CV of Miss Rabonti. So please wait. Okay, okay. Hopefully it's clear now for everyone. So for our second speaker is Miss Shrabonti Chatterjee, and she is a PhD student in molecular biology in Visva-Bharati University, India. And he had several publications. It's also about the virus, I see here. It's also about the cancer biology. He also had many award and scholarship, and now her dissertation project is about the amelioration of high-fat diet, including stress in liver tissue of rat. That's really interesting. But today, I think she will talk about the COVID-19. Is that correct,
Yeah.
Miss Rabunti?
Yeah, I will be talking about my one of the published paper.
Wow,
So I'm
that's really
sharing my screen.
sure, sure. Okay, please start your presentation, and the presentation will be 40 minutes with 5 minutes of question and answer. So for anyone who had the question, please write your question in the chat box, or you can do the raise hand in the Q&A session. Okay, Miss Rabonti, are you ready?
Yeah, I am ready now.
Yeah.
So, first of all, I would like to thank Dr. Arif and all the organizing members for giving me this opportunity. So today I will be talking about one of the like in silico work that we have done with the viroporin. And so this is the title of the paper that I will be discussing today. That is COVID-19 ORF3A viroporin influenced common and unique cellular signaling cascade in lung, heart, and brain choroid plexus organoid with additional enrichment microRNA network analysis for lung and the brain tissue. We all know that like COVID-19, it occurred for a long duration, and in that duration, we saw that different variants were coming again and again. And it was because of the mutations occurring in some of the essential proteins. So ORF3A is one of the accessory proteins of COVID-19, through which it actually it's very important for entry in the host. So we were only discussing the ORF3A protein for this paper. So like people can be, because we know that the spike proteins and all other essential proteins, they're already, it's a lot been discussed and studied upon. So we thought of like exploring the ORF3A protein in
I'm
the
sorry, Miss Shrabonti. In here, we see that the slide is still in the cover slide. Is that correct?
yeah. So I'm moving to the next slide now.
Okay, so sorry. Yeah, please
Yeah,
continue.
it's so general, we see that before also, when it was SARS-CoV and the beta-CoV, that is the first initial COVID that occurred, the first one, in that also viroporins were present, and they play an important role in the viral entry in the host. So in general, viroporins are the proteins that will be actually responsible for creating small channels in the membrane. And then they can be like varying the altering function of ions, that is hydrogen and potassium, calcium, and specifically the chloride ions are mostly affected, the chloride and the calcium ions, if we go through the literature. And its function can be varying that depending on like which virus family it is. It's not only that it's present in the like coronavirus, in other viruses also it is present. And some of the functions that I have mentioned, that it aids in the cell entry, cell lysis, then particle production of the viruses, and it also causes like pathogenesis, like the interferon production, then cytokine production, and also the cytokine storm that it was occurring in the patients. That for that also these viroporins are responsible. So here let's see that where actually the ORF3A, that is the viroporin of COVID-19, is present. So here we see the genomic like sequence or the genome of the SARS-CoV-2. So we see that ORF1A and 1B, these are the non-structural protein part, and rest we have the structural protein part. So in this non-structural protein part, we are also studying now. Like recently, you can see that there is one NSP6 which is occurring in the non-structural protein part. So that also we have studied it later. But for this paper, I am discussing only the ORF3A that is actually coming under the structural and the accessory protein. So these, the main reason behind its like study is because its mutation is responsible for actually leading to the variant production, that is the Omicron and other variants which were occurring. So we saw that there are mutation in the ORF3A, and it can be causing it can be one of the reason why the more pathogenesis was occurring in the later variants. So I will be discussing about the viroporin ORF3A in detail in my next part. So why ORF3A? So this is only with related to the SARS-CoV-2. What is the main functioning of the ORF3A it is causing? So it is reported to be its structure is already done, and it is already present in the database. So we can see that it is actually a 62 kilometer dimer and 124 kilodalton tetramer, and it causes inflammatory responses in the host, and it also helps in the replication of the virus. So it's unlike the that is a SARS-CoV. Like in SARS-CoV also ORF3A is present, but in the SARS-CoV-2 we saw that it's mentioned that it is promoting the lysosomal exocytosis mediated viral egress. It blocks the autolysosome formation, and it interferes the snare complex. So snare complex, we know that it is responsible for like vesicle formation and the transportation. So mainly that functioning is altered because of the ORF3A, and its mutation was actually increasing the mortality rate that is already studied. So we observed that it is also observed that ORF3A viroporin has a relatively weak pro-apoptotic activity compared to the ORF3 viroporin of the SARS-CoV when it is expressed in the cell line. And one of the major function that is observed is it induces the NLRP3 mediated inflammatory response. That is actually it is causing stress to the ER, and then further in like protein folding and everything is going to be disturbed. So here in this study, we are not directly looking for the effect of ORF3 in overall host because it is already been studied. Now we see that in long-term COVID also, and in different patients, there were few tissues which were getting actually too much severe infection was caused, like in the brain, lung, and then in the heart and in intestine and liver also. So we are not able to do all the tissues, but we are doing a tissue specific like pathogenesis, what can be occurring because of the viroporin. And here we are studying only 3 tissue, that is the brain, heart, and lung. So before going to like tissue specific effect, I want to mention here about one of the paper in which they have like experimentally with the help of pull down assay. In the Gordon et al. Paper, they already mentioned what are the different structural and all the non-structural protein also in the host, what are the different target. Like these are the human proteins, and this is the viral protein in red one in between. So it's just the ORF3 I'm working with. So I am presenting here what are the interacting partners of ORF3A. But in the Gordon et al. Paper, like if someone wants to work in like all the non-accessory protein also, which are still not studied properly, like experimentally, so that can be explored also. So we have all the non-accessory protein which is provided in this paper. All the interacting partners are there. So if someone want to like study more, so these interacting partners can be studied in depth with the help of the in silico studies. So we were concentrating on the ORF3A. So these are the 8 protein partners in the human or the host, which is already been studied. And so we are taking all these 8 proteins, that is the interacting partner of ORF3A. Now we are further delineating the protein partners of all these 8 in a tissue specific manner. So in order to get the tissue specific genes related to ORF3A, now what we are doing, we are finding out different literature studies where already different tissues have been studied, and there is RNA-seq data or some other like genome sequencing data is present or not. We searched for that. So we found there for brain, for lung, as well as for heart. So in brain, it was not the complete brain, it was the choroid plexus organoid. So choroid plexus organoid is actually it's actually the cells which are networking the major ventricles of the brain, and it is also present, these cells are also present in the cerebrospinal fluid. So we can be like studying which pathways or which signalings can be affected, and whether the virus is also crossing the blood-brain barrier or not, because this also it helps to study with the help of choroid plexus organoid. So we have 3 data sets, that is for brain and 1 for lung. So there are 3 different papers actually in which they have studied the sequence. They have done the sequencing, and they are finding out which genes are actually getting differentially regulated after the COVID infection in the brain, lung, and exclusively in the cardiomyocyte. So we are taking those differentially regulated gene sets, and then we are further selecting only those genes which are having like significantly regulation, like either up or down regulated. That was more than I think 2.5 log fold change regulation we are selecting. And of course the p-value was the significant ones, because there are a lot of genes which are also non-significant in the data, so we have to eliminate those things. And then we are selecting all these three data sets. And now we have IID database, that is integrated interaction database, in which we can get like tissue specific genes from the host. That is in human, we know that there are different like differentially regulated genes which will be only expressing in different tissues, because all the genes are present in all tissue, but they're not going to be like expressing every time, or it's only selective genes which will be expressing in different tissues at different time. So this is the brain, heart and lung tissue. So in IID, like I showed the previous slide, that we have these 8 interacting partners of the ORF3A. Now we are finding out the further interacting partners of those 8 genes in the IID. Now we are just giving input of those 8 genes in the IID, and then in the tissue specific manner we are further finding out their interaction. So one by one we can go, that in the brain, if you like give the input of those genes, then so brain tissue specific interacting partners you will get, then in heart, and then again in lung. In that way we are collecting all the 3 different data sets. Now what we have to do, so these were already published DEGs, what we have from 3 different RNA-seq data, that is the patients, in the COVID patients, which it is getting expressed. And this is from the IID, that is in the normal host, what are the different tissue-specific genes present. Now we are just finding out the common ones. So in this way, we are finding out the probable genes which are actually getting regulated or maybe altered because of the ORF3A in different tissue. Now we are finding out which genes can be the most regulatory one. So for that, this I already showed, that is this PPI network we will get from the IID, and then we are going to be further finding out the hub genes. So when we are getting the genes from the IID as well as from the DEGs, now we are finding out the common genes, that is the gene sets which we are further working on. That is the genes which can be altered because of the ORF3 viroporin in the humans. Now when we have those tissue specific genes, now one like reason behind a study of this was also because of the drug targets. We know that in COVID-19 different tissue are getting affected. Now in case if we can have an opportunity to have only one drug, and that can be like targeting all the 3 tissue, that will be a very like wonderful thing. So for that we have to actually find out the common genes in all the 3 tissue. That we did it. So that is actually, first we have to go to the string, and in string I created a network of all the common genes in 3 tissue. And then with the help of Cytoscape, in Cytoscape there is a CytoHubba plugin. So there we can find out the top 10 hub genes. So I'm fine. We can see that here the Notch1, HRS, STAT3, ESR1 and PCAM1. These are the top 5 hub genes. That is with the most dark colored shade. That is the most regulatory ones. That is the most important in this network. So we can like people do these type of screening before they find go for some marker genes for which they target any drugs. So these, the Notch1, HRS and STAT3, these are like the important genes in this whole network, that is the common genes which can be affected in the tissue, that is brain, heart and lung, and which can be targeted by a single drug. If there it is, if people can do further this type of work, it will be helpful for them. So finding out the hub genes, and after finding out the hub genes, I also did the cluster analysis of this whole protein network that I got from the string. So now with the help of the M-code plugin again in Cytoscape, we can find out whether different cluster proteins are present or not in the whole big network of the protein. So we found that there are some seed proteins in each cluster, and these seed protein are also important like protein partners which can be used for the drug targeting. So these are like MK6, Ki67, then Esrin, these are in the cluster 1. And then we in cluster 2, that is this UV radiation resistant associated gene and ESR. Again, it is present in cluster 2, and this tectonic family member 2 is present in the cluster 3. And in cluster 4, we have this monooxygenase gene. And in cluster 5, again, we have the EZR. So when we are studying in the M code, all these clusters, so there are generally very different parameters that we have to look while we are performing this analysis. So these things are already mentioned in detail in my paper. So I have not actually mentioned everything like in depth. So moving on to the next part, after we find out the cluster. So this was the common proteins which I took from the brain, heart, and lung. Now next, if we want to see whether the unique proteins which are getting affected in the brain, heart, and lung, like if we want to go to the patient specific management, like if someone some patient, if only the brain is getting affected or heart is only getting affected, so for that we also have to study the unique genes or the pathways which are getting affected. So this is the Venn diagram where we have you can see that different genes and the number of genes which are common in all the three tissue as well as the exclusively in different tissue. And after finding out these genes, we perform gene ontology enrichment analysis. So this gene ontology enrichment analysis can be done with the cluster profiler package in R. So we only have to give the input of the genes, and then in the Homo sapiens package, it will go to the Homo sapiens database, and it will collect all the pathways which can be affected because of these genes. So this is the dot plot of only this 44 genes, that is the common ones. And we see that the red dots are the mostly, highly ranked, that is with a highly significant value. And here we see that these are the count of the gene, that is 5, 10, 15. So the count of the gene means maybe this pathway, that is if I go to the maximum regulated one, that is significant one, that is 0.00. So here I see that it is the vesicle organization that is getting highly affected, or that this pathway is highly getting affected. And this is actually controlled by, I think there is some problem here. It should be more than 15 actually. So it is like more than 15 number of genes are actually controlling this pathway. So in this way, we get our idea what all pathways can be affected, and these pathways can be also further targeted for the. Now, after finding out the common pathways, now we will studied the upregulated as well as the downregulated pathways. So how we got these two different up and down regulated pathway, because in the tissue specific, I already showed here, that is in the Venn diagram, I have 21 DEGs which are getting affected. So from DEG, we also further delineated that which one are upregulated and which one are downregulated. So, and then further, we make two subset of the genes, and then you give as an input to the R, and then it will, we are further finding out which upregulated pathways can be there in the brain. And these are again the downregulated pathways. And here we see that the smoothened signaling pathway will be getting downregulated. And here we see some the phosphorus metabolic processes can be getting upregulated. So similarly in the brain, as we have done, similarly we have done in the heart as well as in the lung. So we see that in the heart, I want to show that here were some of the pathways which were like coming from the calcium related pathways. So here I see. Oh, that will be in the, okay, that was in the common one or the unique one. I don't know. I don't remember, but the calcium pathways were getting affected in the heart. And yeah, here it is, negative regulation of the calcium ion transport. So this is one of the pathways that we already got in our analysis, and it is already reported that this calcium binding or the calcium ion transport are actually getting blocked because of the increased infection in the COVID-19 patient. And now there are some patients which are also taking calcium channel blockers in which their hypertension is there. So people who are already taking calcium ion channel blockers and now they're also getting affected by the COVID, they saw that there was too much this calcium because of the increased calcium blockage of because of this COVID. The patients were like there was a death of the patient or they were going to a very severe condition. So this is one of the finding which we got that is already like reported in the literature, that there are many patients who were taking the calcium ion channel blockers, and it was leading to the severe they were leading to severe hospitalization after the COVID infection. And one of the reason was that we predict is because of the down regulation of the calcium ion transport because of COVID infection. So after finding out the pathways, next part, what we also studied in our this paper, that we are also trying to look for the microRNAs. So why we are also looking for the microRNA aspect? Because it is not properly studied that what are the role of microRNAs in the COVID infection. So suppose there we got many DEGs because of the infection which are getting altered in the tissue, what I showed in the previous slide. Now those DEGs can be actually regulated by the microRNA. It can be possible. So we are trying to find out whether these genes are getting affected by the microRNAs or not. So for that, we are only taking the first initial 8 proteins that I showed in the first 2 slides, that is the second slide, the 8 protein which are already been shown to be interacting with the ORF3A, that is by the pull down assay experimentally. So these are the 8 proteins. And then in the MIRNet, it is one of the software which not software, it's program. Like normally we can access it in the MIRNet. We can find out the microRNA target of these protein. And then these microRNA targets, we further studied whether these microRNA are actually present in the COVID reported studies of COVID patient or not. So initially, I'm showing that how many microRNAs we got with the maximum degree and betweenness. So whenever we get microRNA target, we have some degrees. So it is on the basis of that degree and betweenness only we can tell these microRNA are important or not. So this is these 3 microRNAs I can show, that is I have HSA MIR13P, that is HSA MIR13P, this one, and the big black node, that is green node, that is actually having degree 7. So it is interacting with many different proteins. Then we have HSA MIR1243P, that is this one, HSA. So we can see that it is actually having connection with many different proteins. So maybe it is controlling these proteins, and finally at the infection level or in the patient also. So these microRNA, they act as the hubs and they play a very important role in the interaction network for their this degree and betweenness. So one of the shortcoming was there, that is in MIRNet, we only have the data for or the microRNAs for only two tissue, that is the brain and lung. So this was for brain and this is for the lung. What we did in heart, because the database is not yet present in the MIRNet, so we can perform the probable microRNAs which can be altered in the heart. So after that, so we are also finding out the biological processes that also can be known with the help of the MIRNet enrichment analysis. In the MIRNet itself, there is an enrichment analysis by which we can find out which pathways can be affected overall because of this microRNA and targeting these 8 proteins. So these are the some of the common like pathways, like in both brain and lung, which we got these pathways, that is the NF-kappa B induced immune signaling cascade was getting affected, then cell migration regulation, then transcription factors, then endothelial cell proliferation, protein, then cytoskeleton rearrangement, all these can be getting affected. And this is the unique ones. So this previously was the common pathways which could be affecting in both the lung and the brain. And this is the like unique pathways which I have mentioned here. Now, next to further validate whether these microRNAs what we got, whether they are actually playing any important role in the COVID or not. For that, we found out the literatures which are actually um, reporting that experimentally there are various microRNAs which are found to be getting affected in the COVID patient, and those are actually not the tissue specific but the circulating microRNAs. So we got those circulating microRNAs only. So here I'm showing it, that is these 1, 2, 3, 4, yeah, these 4 circulating microRNAs were actually common with our analysis. So there was a huge 2-3 list of data, that is I have mentioned in everything in detail in my paper. So from those data sets, we find out the common ones, microRNA, which are present in our data also, and it is also being reported to be circulating in COVID patients. So we found that these are the 5 circulating microRNA that is actually altered in the COVID patient, and we also see to be getting affected, or in our analysis we got these 4 microRNAs, and it is in the brain only, and this is for the lung. So among the circulating microRNA, we got only 1 microRNA matching with our analysis in the lung, and that was HSA MER 142-3P. And in the brain, I already mentioned these one, and these 4 microRNA are actually targeting the STAT3 and the Welch domain repeat containing protein 6. So if someone want like further, they can be finding out the whether these microRNA are actually in playing any role in the tissue specific manner or not, because we are only like finding out the probable like outcome of the microRNA, but we don't know actually whether they are actually causing any like upregulation or downregulation of these genes. So this is the microRNA part, what we found out. And this microRNA can also be used for the drug targeting if someone who want to do it. So we are actually not performing any drug target analysis in this paper, but in my next work, I did drug targeting also. But here we have not performed any such like probable drugs which could be like acting on both the proteins or as well as on the microRNA. We have not seen. So this is the overall view that what all we have done. So this is the that is in the heart, brain, and the we have lung. So we are finding out the ORF3A protein, if it is infecting the tissue, or it is like entering the tissue, that when what are the differentially regulated genes which will be affected, and then further finding out the functional analysis in and the microRNA also. And these are the pathways which are like prominent pathways which could be getting affected. Similarly, in the heart also we see the if ORF3A is infecting, or it is entering the heart tissue, what all pathways it can be affected. And one of the major thing which I already mentioned is the calcium ion channel blockers, which already has been reported to being it's going to be increasing the activity of those calcium channel blocker. And then so this part should be it should be studied in depth, because all these things are causing in long term, if someone is seeing that after COVID many tissues are actually getting affected, and those it is not getting recovered like in long term. So these things can be kept in mind, and further drug target or therapeutic aspect people can study. So this is the the DOI of my paper. And thank you all. And these are I already mentioned these things in my when I was explaining. So one of the thing is caveolin 3, which I was telling, that is CAV3 is down regulated in all the three, and this caveolin 3 is actually the protein which is getting affected because of the ORF3A in heart, and it can be actually increasing the calcium channel. It's decreasing the calcium ion transport, and then leading to the too much decreased activity of the calcium ion channel blockers, because of which patients can be it can be having a severe effect. And another gene is TMBIM6. It is also observed to be downregulated in SARS-CoV-2 infected cells, which may prompt the calcium ion disorder in the cell. So this is one again more gene that has been reported, and this is the what we also got to be affected in our analysis. We got this gene that is actually causing it is down regulated, and it is also altering the calcium ion activity in the heart. And some of the pathways we got is the macroautophagy in the brain, heart, and lung, the regulation of the autophagy, and this was common in all the three tissue, this pathway. And it is also being reported that in patients which are severely infected, they have the impairment of the autophagy markers. So this is also showing alignment with our analysis. And this Notch1 signaling is also getting affected, and it is one of the hub gene what we got in our analysis in all the three tissue. It is one of the major. I showed it was in red mark, and it was one of the important hub gene which is getting affected. And also in SARS-CoV-2 patient, the Notch signaling is actually getting altered. So these are some of the conclusions of my work. And thank you all for listening patiently, and if I'm open for question and answers.
Thank you so much, Ms. Shrabonti, for the wonderful talk about the finding of phytoprotein. And we finally know the interaction that related to various organs, right? Including the brain, heart, and heart. And also lung. It's completed with mRNA data, right?
Yeah.
Okay. So now we are going to the Q&A session. So if anyone had a question, please do raise your hand, or you can write your question in the chat box. And in here, we already have two questions, Ms. Shrabonti. It's from Mr. Zahid Salim from University of Sahiwal, Pakistan. He asks about could the identified mRNA network be used as the therapeutic target or biomarkers? How do you think?
Yeah, it's already, I mentioned that
Mm.
microRNAs can be used as a suitable biomarker. So I told that specifically in tissue, it is not yet reported in literature. That is the experimental thing. Like before confirming, we have to like go through that phase before we tell, yeah, it can be used as a biomarker. So, but there are circulating microRNAs which has been reported. So among those circulating microRNA, we got 4 microRNA that can be, yeah, aligning, and we can study further for the tissue specific aspect, like experimentally. And then we can say that, yeah, these microRNAs can be used as a biomarker for this tissue and this tissue, like what I have mentioned in my study. And for time being, I have not gone through like recently if there are any tissue specific biomarkers of microRNA present. When I was doing this work at that time, there was no like as such biomarkers in the microRNA. But yeah, these microRNAs we can further study for the drug targets. These are like one of the good important aspect for therapeutics.
Right, right. It still needs the further confirmation using in vitro, right?
Yeah.
Yeah, it's wonderful finding. Okay, so in here I found the further question from Mr. Zahid Salim. It's about the criteria of the microRNA that may be suitable become the biomarker in the context of COVID-19. And is there any known microRNA that already established as biomarker in COVID-19 patient that maybe could align with this study's finding?
No, till now there are no biomarkers which are, but yeah, I showed that few microRNAs are aligning with our study finding, but they are not the biomarkers. There are just like microRNAs which we can say it could be targeting our genes. I mean, which genes it could be targeting, that is also one of our finding. But whether it is used as biomarker till now, it is not known. In like literature studies also, I think it's not known.
I see, I see. Thank you so much.
Yeah.
Okay, so we actually still have time here. Maybe I just this is from my personally like just curious about your research because commonly I also work that similar study about protein interaction, and I just curious when you mentioned about the organ like from lung, heart, and also from the brain.
Yeah.
In that specific organ, do you also check the protein from the specific area of the organ? For example, if in the brain had like many part, right? Do
Mm-hmm.
you also identify to that?
No, actually, it was not possible because in the RNA-seq data, which is, or the transcriptomics data, which is already present, there are some limitation. I mean, in this study also, I am not taking the brain as such. I already mentioned it is a choroid plexus study because the complete brain, I think till now there is no report. It was the choroid plexus which people used for the transcriptomics. And for heart also, there are cardiomyocyte cells, which is it is present. It's not the complete heart maybe. But for lung, yeah, in the lung database, when if you go to the transcriptomic data set, there were different cells it was present. So if you further want to delineate, then we can go to the specific cell type study also, like if we want to do. But in this study, I have not done. So
Thank
it's
you so
depend
much.
on the transcriptomic study, I mean, data what we have in
Yeah,
the
yeah, that's
database.
correct. The database is really essential,
Yeah,
right?
yeah.
Yeah. Thank you so much, Miss Shrabonti, for the wonderful
Yes.
talk. Maybe after this we can have a discussion more, okay?
Yeah, yeah,
Thank you
sure.
so much. Thank you so much,
Welcome.
Miss
Bye.
Shrabonti. Thank you.
Bye.
So, everyone, so now we are heading for the third session with the third speaker. So in here, I'm honored for welcoming Dr. Kavitha Kandiah. She is
Yes.
the associate professor at K.S. Rangasamy College of Technology, India, and Currently, she also become the visiting researcher at BRIN Indonesia. Wow. Hi,
Hi,
good
welcome.
morning. Good morning,
Yeah, good
Dr.
afternoon here.
Kavitha.
Yeah.
How are you? Are you stay in India or in Indonesia now?
In Indonesia.
Ah, so it's already afternoon.
Yeah.
Yeah, yeah. How is that? Is that
Yeah, it's
great?
nice.
Everything there?
All the people are very nice here.
Thank you
Very
so much.
smiling.
Thank you so much. So, Dr. Kavitha, before we are going to start the presentation, I
Yeah.
will share about the CV of
Okay.
yours in
Yes.
here just to let anyone know. So in here, I will share maybe in here. Okay, hopefully that everyone still can see here clearly. So this is the detailed information about the Dr. Kavitha Kandiah. Previously, she is the associate professor in Department of Biotechnology in KSRCT India and also become the visiting researcher in BRIN Indonesia. She had focused for the biomaterials and also the nanomaterials that are going to be her presentation from after this. And she graduated from the PhD from the same university, KS Rangasamy College of Technology, India. She had many awards and honors, and also she had about 28 international journal that had focused for the nanomaterial. So it's really interesting when we can heard uh, about the nanomaterial from Dr. Kavitha. So, Dr. Kavitha, are you
Yes.
ready?
Yeah. Shall I share my screen?
Yes, please.
Okay.
I hope that it could easily connect it.
Is it visible?
Yes, it's already clear. So now the time is yours. Thank
Yeah.
you.
Thank you. Okay. Good afternoon, everyone. Welcome to 19th International Webinar Series. I'm Dr. Kavitha from KS Rangasamy College of Technology. Am I clear? Am I audible?
Yeah, it's clear, but I think for the presentation, it's a kind of freeze. Could you please move to the next slide, Dr. Kavitha?
Yeah. I'm in this first slide only.
Ah, okay. Thank you.
Okay, I'm working in KS Rangasamy College of Technology, Tamil Nadu, India. I had the enriching opportunity to work in Indonesia for 3 months as a visiting researcher, which gently and greatly broadened my global perspective and fostered the valuable international collaboration. During this time, I can able to share as well as gain the knowledge within a vibrant research community. It is a truly an honor to be here with you today to explore the insight facts about the biomimic biomaterials. India is my beloved country, which incredibly diversity with 28 different states, more than 10 religions, 22 official languages and 1,500 unofficial languages spoken across the country with unity. This rich cultural fabric is one of our strengths and greatest strength. I come from Tamil Nadu, a southern state known for its deep-rooted heritage, and I am currently working in KS Rangasamy College. It was located in the blue color bin.
Hi,
This
Dr.
one's-
Kavitha. I'm
Yeah.
sorry for to distract your presentation, but in here, your slide it's not presented yet, so we still can see your window for the PPT, but not presented yet.
Okay, wait, I try.
Or maybe try to stop the share screen first and then present the slide and then try to share screen again.
Yeah. Is it okay? Shall I present like this?
Yes, but it's like for the window of the PPT, it's not presented yet.
Yeah, but if I present, it was not shown in the
Oh, I see. Okay, so if you comfortable for using the window like this, so it's okay because we still can see clearly.
Okay.
Okay. Thank you so
Okay.
much. Yeah, it's clear. Okay.
Okay. Okay. This is India is actively promoting research, particularly in bio-related sciences, to support the emerging researchers. The government offers various grants and fellowship through different agencies like UGC, DST, DMIT, and ICMR. Like that, many agencies are offering the fellowships. They are highlighted fellowships are Ramanujan fellowships, Inspire fellowships, Short of Research Grant, Women Scientists and Vice, and ANRF Yearly Career Research. For these fellowships, India is spending more time and more importance to the upcoming researchers. As like in the next slide. Our nanobiomaterials lab in KSR House equipped state-of-the-art instrumentation of advanced materials research, including 3D printer for scaffold fabrication and harness testing, SEM, AFM, FDR, TGA, BET, non-indentation, like all the characterization techniques we have. Also for synthesis, sonicator, hydrothermal spray pyrolysis, and electrochemical deposition, all the instruments are available for nanosynthesis also. These sophisticated facilities enable us to obtain comprehensive and reliable results, driving innovation and excellence in our research. Okay, now we are coming to the point that is biomimicry is the art of copying nature's solution. It's nothing but copying nature for existing problems. Nature has answers to all existing and future challenges. For that, we have to understand and adopt the natural process and design. Then only we can able to develop a groundbreaking innovation such as wind turbines inspired by dolphins and termite mound inspired sustainable buildings and noise reducing technologies inspired by kingfisher beaks, and like that all the existing sustainable innovations are inspired by nature's only. Let's explore more examples of human invention inspired by natural designs. These are the examples we can see one by one after the slides. Okay. Do we need biomimicry in biomaterials, especially in nanobiomaterials? The answer is clear and strong. That is yes. Nature has uh, perfect. Nature has the solution, perfect solution over million of years of the evaluation. By mimicking the biological system at the nanoscale, we can develop advanced biomaterials with enhanced biocompatibility, superior mechanical properties, and improved sustainability. I think all are aware about nanoparticles. It's nothing but 10 power minus 9 meter in dimension, smaller than micrometer, that is virus, DNA, proteins, all are comes under nano. These bio-inspired biomaterials in nanoscale are revolutionary one nowadays for biomedical application, especially targeted drug delivery to tissue engineering, offering innovative solution to our most precising healthcare challenges through the smart materials combination and nature-inspired designs. Nature is also offering wisdom and guidance as like our ancient scriptures like Mahabharatam, Quran and Bible. However, unlocking its secrets requires scientific inquiry beyond mere observation. The lizard's ability to climb walls without falling down is due to its unique food structure and adhesion mechanism. We have to admire how the hair-like structures will give adhesion for this lizard to climbing on the top of the surface of the wall. By inspiring this mechanism, some climbing robots were prepared, and industrial robots to hold the things, and medical adhesive bandages are also developed by inspiring these lizards. The lotus leaf is a remarkable one. This is it having remarkable nanostructure, showcases super hydrophobic properties, enabling repel the water from its surface, taking dirt and contaminants with them. This nature self-cleaning mechanism has inspired a range of innovative applications, including self-cleaning paints. Right? Yeah. Self-cleaning paints and coatings, water repellent glasses and windows, water resistant coating for various industries, and low maintenance surface for building and infrastructures. Like that, top of the covering all the messy things, we can use this self-cleaning coating. Next one, butterfly wings. This is own their vibrant color to unique photonic nanostructure that reflects specific wavelength of light. This optical mechanism has inspired innovations like anti-counterfeiting technologies for currency and IDs, optical sensors, and advanced authentication systems. By inspiring the butterfly effect, a lot of innovations are now arrived. Next one, shark skin have a unique structure, exhibits antibacterial and antifouling property by repelling the gram-negative bacteria and preventing the biofilm formation. This nature defense mechanism has inspired innovation like hospital surface coating and instrumentation where we are using in the operations or any other medical related things. We can coat this nanoparticles over the surface of the instruments. For that, we can prevent the biofilm formation. Implant and cathode coatings and medical instruments is coated by these type of nanoparticles. Next, pine cones, which is inspired by the it is used for passive ventilation system in buildings as well as moisture regulating fabrics. Pine cone outer layer will be rigid and inner layer will be absorb the water and swell and maintain the humidity. Hence, this mechanism is copied and applied in the fabrics we are using now. Next one. Human eye is ability to handle the sunlight. So researchers have developed sunscreen inspired by its natural mechanism. Anti-reflective properties in sunscreen provide effective protection, showcasing nature's influence on the innovative solution. Next one, some other interesting innovations like plant-based volatiles inspired packaging sachets that extend the food freshness by mimicking the natural preservation mechanism. Another one, Secmo sheet inspired ceiling fans with aerodynamically optimized blades for silent and energy efficient operations. Next one, anhydrobiotic organism-based technologies that enable stable COVID-19 vaccine storage without extreme refrigeration. Next one is virus-like nano cages engineered through the DNA, origami, and 3D printing for previous drug delivery. Nature is the ultimate innovator, and biomimic biomaterials in medicine prove it. Using the following examples like the hydrogels copying extracellular matrix, which is present in our body for tissue engineering application, and starch skin surfaces fight the bacteria without any chemicals. Spider-like polymers heal the wounds with super strength, and it will also act as a antibacterial activity by if we're refining the structure with some other composites, we can act as that spider's fiber act as a antibacterial agent also. Okay. Next one is artificial cell and biohybrid system. Engineered artificial cells mimic the natural cellular functions, enabling target drug delivery and controlled biomineralization for bone regeneration. Bio-inspired optical and neural technologies. Okay, neural technologies. For this, human smell and taste. Here, okay. Human smell and taste taste buds system inspired and give the inspired artificial odor sensing machines and smart signal processing for the medical diagnosis. And these are the current challenges and future trends. Despite their immense potential of biomimic biomaterials, face several challenges. One major issue is replicating the complexity of the natural structures, which is not easy with the current technology. Fabrication at scale remains difficult, limiting the commercial applications. Additionally, high production cost and strict regulatory frameworks hurdles to widespread adoption. Addressing these challenges is the key to translating biomimic innovations from lab to life. Looking ahead, a future of biomimic biomaterial is incredibly promising. We are moving towards smart biomaterials that can respond to stimuli like pH or temperature, enhancing their functionality in real time. Biohybrid robots combining living tissue with synthetic system are also emerging as a game changer. Moreover, AI-assisted biomimic designs is accelerating innovation by mimicking mimic the nature more efficiently. These advancements hold the potential to revolutionizing healthcare, robotics, and material science. Okay, now we can move to. Am I audible?
Yes, it's clear.
Okay,
Dr.
okay.
Kavitha.
Okay. Now we can move to nanoparticles related one. That is, there are two type of nanoparticles. That is organic and inorganic. I think all aware of this one. Other than the carbon, all other metals and metal oxides belongs to the inorganic nanocomposites. Why this nano is special means it's having unique properties. That is the main property is surface to volume ratio. We can by using that surface to volume ratio and other shape and size, it will if we prepare the particles according to the requirement, it will change the property. So it will roll on all the fields now very efficiently, and depending upon the methods, what methods we are using, it was categorized in three category. First one is physical method, another one is chemical, another one is biological method. Similarly, there are two different methods is also there, that is top down and bottom up method. In top down method, from the bulk materials to nano, we will reduce using some physical methods. In bottom up, from the bottom means it is atom by atom synthesis will be followed in bottom up synthesis. These are the characterization techniques we are using for nanoparticles: XRD, FECM, FTR, and HR10, BET, and UV spec and photoluminescence. These are the major equipments we are using for characterize the nanoparticles. These are the fields the nanoparticles and nanobiomaterials is used now. And this is my work. What we will mimic the biomaterials. This is the first material. This is a TiO2 nanocomposites for mimicking bone cements. Biomaterials are intended to interact with the living system. We choose TiO2 nanoparticles as a base and combining some polymers other than and carbon materials to support this bone mimic cements. And finally conclude according to our all the characterization and biological results. TiO2 and chondroitin 4 sulfate polymers have a best among the prepared one for orthopedic applications. However, it's having some variations in all other characterization and biological application. This will be concluded. Next one is TiO2 nanocomposites coating on Ti alloy. For load-bearing application, we are using very hard composites or a hard alloy for as a implant. So, but that alloys have some difficult, some disadvantages. That is, it will form biofilm on the top of the layer, top of the implant. So we can use TiO2 nanocomposites as a coating to prevent the biofilm formation. Next work is biosynthesis of graphene oxide. Actually, this we can use many precursors. One is jujube and mushroom and some other grasses of our Indian grasses for preparing a graphene oxide to reduced graphene oxide. We use this plant source as well as microbial source for reducing the graphene to graphene oxide to reduced graphene oxide, and we analyzed in animal all the after preliminary studies in cell lines and biological SBF fluid, everything. We can move on to animals and concluded that these graphene oxide is non-toxic one, and we can go further for the composite preparation and bone replacement applications. Based on the above preparation, we prepared graphene oxide, HAP, and extracellular matrix polymer composites, and found good bone implantation and bone growth. Next one is biosynthesis of nano magnesium oxide from the rutile soil, which was isolated from Salem, as well as soil and soil isolated actinobacteria act as a reducing agent in for MGO synthesis. This nano MGO is used as a nano mineral for many other applications, especially in animal nutrition, as well as bone related growths. This one is carbon quantum dot and activated carbon from multiple sources. This carbon derivatives are used for energy storage application as well as animal nutrition and fertilizer applications. Thanks to the BRIN for offering visiting researcher position and giving the place to do the research, and as well as I'm sharing the knowledge along with gaining the knowledge. Here in BRIN, I have focused prebiotic and probiotic combinations that is called symbiotics. This symbiotics is useful for animal feed as well as agriculture. For sustainable and reusable purpose, we are isolating prebiotics and probiotics from the palm waste. Yes, it is not to glorify, but ancient Indians, especially our Tamil forefathers, lived in perfect harmony with nature, leading healthy and fulfilling lives without relying on the technological advancements. So we can able to adopt and understand the nature. Thereby, we can invent many groundbreaking innovations and technologies nowadays. Okay, these are my highlighted publications with cumulative impact factors of 120. And I'm concluding that. Thank you for VRCBB and Mohammed Ansari for giving this opportunity. Thank you. Thank you to all.
Thank you so much, Dr. Kavitha. That's a really insightful talk. So in here, basically, we can get the inspiration and also the idea for developing the drug or nanomaterial from the nature, right?
Yeah.
That's really interesting. So for everyone, so we are now going for the Q&A session. So for anyone that had the question, please do raise your hand or you can write your questions in the chat box. And in here we have one that raised the hand. This is Miss or Mr. Dioang. Hello, Miss or Mr. Dioang. Can you hear me? Hello. Hello, Mr. Dioang. I saw that you raised your hand. Okay, while waiting for the Miss or Mr. Dioang, so I just personally interested with this topic, and I want to ask about the biomimetic of the bone repairing. I read here it's about the bone cement development. Is there any toxicity information about that? Because I thought that this bone cement will be developed being like for implant, right? For
Yeah,
to
yeah, yeah,
repair
yeah.
the bone. How about the toxicity for that?
Yeah, without testing the toxicity, we can't able to go for next animal study. After animal studies, only we can go for the implantation. Uh, Usually, we check the preliminary toxicity in the cell line studies.
Okay.
It may be the animal study, animal tissues, or it may be the human tissue. We can retrieve the cells and test it in that. After that, only we can move towards to the in vivo studies. After in vivo studies, only we can go for clinical trials. So after confirming all the toxicity, then only we can move for the application.
Right, right. Because this idea is really applicative, right? If
Yeah,
everything was fine, so we can like going further for the application
yeah,
and for the applicative application, right?
yeah.
That's a really great idea. And how about the main factor that can affect for this bone cement to become like toxic or less toxic? Is there any main factor that can affect for that?
Yeah, if it is adopted to the tissue, osteoblast tissue, it will be no issue. It won't cause any toxicity. But if the adaptation is not there, sometimes it will cause a toxicity and sometimes it will be reject the implantation. If it is rejected, we have to operate again. So that before choosing the material, we have to study in detail about the toxicity.
I see.
Mm, After that, only we can choose the material. And after choosing also, by converting micro to nano, sometimes it will show some toxicity also. So after converting the product into our desired form also, we have to check once. That will be
Right,
give
right.
a
I also
better, yeah.
Yeah, yeah,
Okay,
about
thank
the
you.
material and also maybe the size, that's really essential because if we
Yeah,
are
yeah,
going to apply to the human that yeah, we have to be careful that the human
yeah,
body can consider that
yeah,
material as antigen, right? But
yeah.
yeah, that's a really good idea. Thank you so much, Dr.
Thank
Kavitha.
you.
So I
Thank
think
you.
here there is no question, but if you have any question, please feel free to contact Dr. Kavitha. So the email is there in the
Yeah,
screen. So for anyone that want to do the collaboration and also have a research together or just have a discussion about the nano material, so please feel free to reach the Dr. Kavitha. Thank you so much, Dr. Kavitha, for your
thank you.
share.
Thank you.
Thank you so much. Okay. Now we still have time and we will go for the last speaker. And here it's a really honor for me for welcoming my good friend, Mr. Muhammad Fahmi. Are you there? Hello.
Hello, hello. Can you hear me?
Yeah, it's clear. How are you?
I'm fine, fine. I'm sorry, my camera is not working properly, so you cannot see me right now. I'm so sorry.
Ah, I see. No problem. No problem. As long as you can share the slide, I think everything will be fine.
Okay, we'll try.
Ah, okay. But before you are going to share your slide, let me share about the CV or the detail of Dr. Muhammad Fahmi. And here, I will try to share. Hopefully, everyone can see clearly the CV of Dr. Muhammad Fahmi. He is a bioinformatician at the Bioinnovasi Analitika Indonesia, or Anagenic, and he graduated from Ritsumeikan University, Japan. And previously, he had many experiences to be a researcher in several institutes in Japan, including the Research Institute for Humanity and Nature in Kyoto, and also being a visiting researcher in Ritsumeikan University. And currently, he also published many papers about bioinformatics. So it's really happy that we can hear Dr. Muhammad Fahmi to share his knowledge. So, Dr. Muhammad Fahmi, are you ready?
Yes, thank you so much for
Okay,
your
the time
introduction.
is yours.
Okay, can you see my slide?
Yes, it's clear.
Okay, good afternoon, everyone. First of all, thank you for being here today, and I truly appreciate the opportunity to share with you some of the work I've been doing. Also, a big thanks to the organizer for arranging this session. So today I will present about filter screening in Nextflow, so the process can be fast and more comprehensive. And what you might get today is that an overview of a fast and scalable filter screening workflow, and I'll also break down how Nextflow helps automate and parallelize tasks, making large-scale screenings not only feasible but efficient. Also, I hope that you can grasp a clear learning path and receive inspiration from my talk. And long before the age of high throughput screening and computational docking, humans already turned to nature for healing. One of the earliest known medical documents is the Ebers Papyrus from ancient Egypt. From these papers, they describe over 700 plant-based remedies, including uh, one of which is willow bark. And today we know that in willow bark it has salicin, and this is the precursor of the aspirin, as we know as we use today as a painkiller. And centuries later, nature continues to inspire life-saving treatment, one of which is the Madagascar periwinkle, Catharanthus roseus. This is used. Uh, this is a key drugs in treating leukemia, which has vinblastine and vincristine. Another one is natural product from animals. This is Polybia paulista wasps, whose venom is thought to destroy cell cancer, and uh, yeah, many natural products comes from uh, has been used as drugs recently. And today we have access to hundreds of millions of chemical structure. For example, in zinc 15 database, there are 750 million processable molecules, and in Chebel database there are around 2.3 billion bioactive compounds, and in drug bank there are more than 15,000 drugs approved and investigational. So there are so many compounds that can be used and can be candidate for drugs. And right now there are around 2,000 to 3,000 small molecules drug approved by major agency like FDA and EMA. So from these many molecules, we need some kind of methods to screen which one that can be used to treat some kind of specific disease. So That's why virtual screening help us to solve on, like to how to efficiently narrow down thousands or millions of compounds to the most promising few. And virtual screening help us to reduce animal and cell-based testing, especially in the early stage, and prioritize candidates that are more likely to bite well, also therapeutic effects to our target disease. And then speed up the discovery process by filtering out weak binders, and most importantly, significantly reduce the cost of experimentation. And this is the common pipelines that we can do in the virtual screening. There are protein and ligand selection, protein ligand preparation, binding site definition, local docking, and evaluation. There are so many steps, and we constantly jump between one file format to another. For example, in virtual screening, there are PDB format, MOL2 format, SDF format, SMARS, and etc. And also, we constantly jump from one tool to another, like Open Bible, PyMol, Autodocs, Mina, if you have done this kind of step. This is the workflow that we usually face in conducting virtual screening. Yes, this is the bioinformatics. Most bioinformatics job is facing this kind of uh, job. Like we are facing many tools, and we are facing many formats, and jump from one tool to another, and that is uh, probably pretty frustrating for us. This is another complex workflow in bioinformatics. For example, in RNA-seq, we usually have a FASTQ input, and then we conduct some kind of basic QC and read streaming using FastQC or Tribomatic software. And then we conduct several steps like contamination check, advanced QC, assembly, and etc. And most of this workflow usually includes so many software and so many formats. One of the very complex pipelines in bioinformatics is annotating genome, parasite genome. So in this pipeline, there are more than 70 process and more than 69 software tools and libraries. So this is the problem that we face, and we need some kind of managers to face this kind of problems. And one of the solution is using workflow manager. So one of the workflow manager is Nextflow. Nextflow is an workflow manager based on Java, and they use Groovy script. But in this workflow manager, you can use pretty any programming language like R, Python, or Bash. You can use it inside this workflow manager. And in this workflow manager, you also can conduct version control. So yeah, so if you build some kind of pipelines, you can put it in the GitHub and then see the progress of it. And you can also combine this workflow with like containers such as Dockers and Singularity and Conda. This case is pretty useful if you transfer your software, so your recipient doesn't need to install all of the software in their own computer. So they can just run the program inside the Docker or Conda. So no need to install so many tools that we need. And in the next flow, you can also run this in your personal computers or so many major platforms like AWS, Google Cloud, Microsoft Azure, and etc. So this is the tools that provided to us to to solve the problem in the bioinformatics pipelining and workflow. So this is the example of managing the challenge of complex bioinformatics workflow. For example, in transcript expression quantification, you have input of ASCII and reference sequence, and usually you run one by one of these a process, or you can create a bash script to make it automatically. Like we have step 1 quality control using MassQC, and then indexation using Salmon, and then so on. So you can get the results. In the traditional pipeline, the process is streamlined like this. But in the Nextflow or Workflow Manager, you can run the process in parallel and with the much more complex or high throughput step. Like for example, it has a scalability, and it meaning it can run many samples in parallel. If you have 1,000 samples to run, so the sample will be run from step 1 to 3. But sometimes if you finish in step 1, it will be directly run in step 2, and some will run in step 1. So like for example, there are 300 samples in step 1 and 700 samples in step 2 running in parallel. So you don't need to finish one step to next step one by one. So it can create an environment to run the process in parallel. And also it has a re-entrancy, meaning if the pipeline crash, so you can resume the exactly where it left off. So it doesn't need to rerun from the beginning. So some data that has been run in step 1 or step 2, they have memory. So if the server crash, they will be run in their exact step. And it also has data provenance, like every step is tracked, and you can make the analysis more reproducible and transparent. And also it is has portability, like the same pipeline runs across a machine, cluster, or even the cloud with no change. In short, Workflow Manager will help you move from a fragile manual scripting to reliable production grade pipelines. For example, this is the task in aligning NGS data in this sample FASTQ to reference FASTA using BWA mem. So this is the command line in the Linux platform, for example. You can run it. And in Nextflow, this is the script that you can write. So this is the script. So it has a process step. It has input and output and script. For script is the process that you want to run, like PWMM with the reference FASTA and sample FASTQ as input, and the output will be this is sample.pem. And you need to describe it, the input, what is the input and what is the output. And from this task composition, Nextflow will run the input output to from one process to another. For example, this output is from a line sample, and if it is connected to the index sample, the output here will be run here in streamline. And one of the best things about using Nextflow is that you are not starting from scratch. There is an amazing community project called NFCore, which provide well-maintained peer-reviewed pipelines for common bioinformatics tasks. Like for example, here is RNA6. This pipeline is available publicly, freely. And for example, in this RNA6, it will automate the process from FASTQ input to so many process like using FastQC to quality control, trim color to trim some index, and then short me RNA to sort the contaminant from this FASTQ data, and it has so many output, like here is GDF FASTA, and then assembling process, and etc. And yeah, this one can be run in Streamline, and you just need the input, and you get so many output from this pipeline, and it is available in this community and of course. But for critical screening, right now, I think some pipeline is still in development, and here we try to also develop our own pipeline using Nextflow for virtual screening. And in this our development process, we have some of the pipeline that we have developed. Like first is Lipinski rule of 5 for the filtration of the compound, and then filtering compound using paints, and then ligand preparation, binding site definition, molecular targeting, and evaluation. We right now developing this pipeline for this virtual screening. And maybe I want to explain a little bit about the Lipinski rule of 5. This is a set of guidelines used to evaluate whether a compound is likely to be orally active in humans. So according to the rule, a compound is likely to have a good absorption and permeability if no more than 5 hydrogen bond donors, no more than 10 hydrogen bond acceptors, or molecular weight is less than 500 Dalton, and log partition coefficient is less than 5. And for filtering unwanted structure, one of which is the PAINS, P-A-I-N-S, pain assay interference compound. So PAINS is a compound that react non-specifically with proteins, so they can bind to so many proteins, and it also interfere with assay detection method like fluorescence and reticorrection. And to avoid wasting time on misleading gates, we also apply PAINS filter with another filter also to subset our substructure, and we remove the unwanted structure. And in this case, we are using library from NPASS and GBIF. So we want to get the natural product derived from plants that is distributed from Indonesia. So we use these two databases. First is GBIF. This one provide the information of species that is distributed in Indonesia. And then the second one is natural product activity and species source database. This one provide information of compound species relationship for bioactive molecules. And then we merge these two information, and then we get like 9,869 unique compound derived from Indonesian flora. So this is the compounds that we use for the virtual screening. And this is the distribution of the pledge orders. Most of them in the fabulous orders, Gentiales, and etc. And from the target here, we use PPOR, or peroxisome proliferator activated receptors. This is nuclear receptors involved in regulating glucose and lipid metabolism, making them highly relevant in diabetes and metabolic disorder treatment. And this one is the target for the drug called pioglitazone. So this one mainly targeting PPR gamma and activating it, and then triggers the expression of these genes, hence increasing the insulin sensitivity in muscle and fat cells, reduce hepatic glucose production, and then improve uh, the lipid profiles. In short, pioglitazone modulates PPR gamma to improve glucose control. And we want to see or seek an alternative compound derived from natural compound from plants in Indonesia. So we conducting the pipeline that we have developed, and then we got more than around 2,662 compound that pass the Lipinski rule of thumb and the paints filtration. And this is the Phylan plots that shows the distribution of docking scores across 5 predicted binding modes for each ligands. So the dot here is the ligands, and there are tiny red dot here, which is the control. And mode 1 here is typically the strongest binding conformation and the most relevant for hit selection. So in this case, our control ligand, which is pioglitazone, so minus 8.59 affinity, which we use as a benchmark. And there are some compound that has more stronger binding affinity than the control. So we select this compound for the next process. And we got like 190 natural compound that has stronger binding affinity than the control. And for the species distribution is again Fabales, in the order Fabales is the most common, and Sapindales, Asterales, and so on. And our pipeline also classify that selected compound for NP classifier. This is a tool that categorizes natural product based on their biosynthetic origin and chemical structure. So from our results, over 100 compounds were classified under Sikimids and phenylpropanoids pathway, and more than 50 compounds fell into the lignans superclass, and then around more than 10 compounds were identified as flavones, and so on. And the next output is the UMAP of the Tanimoto index. So in this case, we projected the index of similarity using Tanimoto index to the UMAP. So we can see the similarity of the structure of our control to the another compound. For example, here our control is pointed in the center of the UMAP, and in this case, the compounds that is closer to this control is more chemically similar, and the farthest is structurally more distinct to this control. And then we pick the 3 most similar and dissimilar compound compared to our controls. We got Vixtramol from this species, which have this affinity, and it has a Tanimoto index to control 0.18. This is still a very low Tanimoto index. So this compound, even though this is the most closer, they are not so similar with our control or the pioglitazone. And the most dissimilar compound is hypuristanol. So this is from Melokia pyramidata, and it has like minus 9.17. And also our pipeline also showing the side by side of the structure of protein and ligand presentation. So this normal. This is the pioglitazone binding to the PPR here. You can see. So this is the stromal, a bit distinct binding, I think. And this is for hypuristanol, also a bit distinct binding. And from the next is so we also have the top 3 most strongest binding ligands. So the top strongest binders is bioinformatics which has less than -10, and it is derived from zeamice or corn. So this compound is, I think, it's a type of glycosylated flavonoid, and it has a link to sugar moiety, and the structure are known for their antioxidant, anti-inflammatory, and sometimes metabolic regulatory activities. And I will show you again. So this is the strongest compound that bind to our target. And this is side-by-side visualization with pioglitazone. So yeah, our pipeline is so one of the key advantage of using workflow manager like TextFlow is the speed, and our results show it clearly on our vertical screening pipeline. It runs 3.1 times faster compared to the executing the same task manually in Jupyter notebook. So in our in my PC, when I run Nextflow, it takes like 1 hour and 40 minutes for like 9,000 compound, and in the Jupyter it takes like 5 hour and 12 minutes. So in summary, we design a reproducible and scalable virtual screening pipeline using Nextflow. It handles filtration, docking, and classification, and similarity analysis in streamlined fashion. And we are still developing this pipeline. We hope we can also conduct another process like another molecular docking, like using GBSA or etc. And in our pipeline outperformed manual approach, achieving 3.1 times faster performance than traditional Jupyter execution, demonstrating that workflow design leads to the smart workflow design leads to the real-world efficiency gains. And yeah, this is my own firm right now. It's called Anagenic. It's based in Indonesia. So we help researchers for bioinformatics supports, and we also provide essential biotech laboratory supplies. If you want a discussion or some kind of help with the bioinformatics, or you need biotech laboratory supplies, uh, uh, you can email me in this email. Yes, I think that's all. Thank you so much for your time.
Thank you very much, Dr. Fahmi, for the great talk about the next flow and anagenic. So in here, we basically know that the next flow is like a workflow manager. So for everyone that concerned in bioinformatics, you must be realized that sometimes we have to run the data with many web server or many tools. So by using this next flow, it's kind of the shortcut. So we just need to set the list of the analysis, and then we set into the next flow, and we just need to submit our sample. So the next flow will manage everything for analysis. Is that correct, Dr. Fahmi?
Yeah, it's correct. Yeah.
It's really, really helpful. Thank you so much. So everyone, here we are going for the Q&A session. So for everyone that had a question, please feel free to raise your hand for directly discuss with us and with Dr. Fahmi. And also if you have any problem, just write your question in the chat box so we can easily answer your question. Okay, while waiting for the participant and the question, I just wondering about the next flow, because in here you mentioned that we can set our next flow, and in the end of the step, we also have the evaluation step, right? So could you please explain about the evaluation? Is that also have the function as the quality control for the analysis or something?
Yes. Basically, for evaluation in Xflow is like we can get the report of the memory that is used in the PC, and then we can get the information of the time that to run one sample, one to another samples. For each sample, we can see the time that takes, and then we can also get the information of the reports of which process that takes so many CPU computational power, so we can evaluate it, and then we can set it to maybe get more computation power, or and so on. And that's kind of evaluation that we can get from Nextflow. So we can adjust our process so we can get the results much more faster.
Wow, that's wonderful. Thank you so much. So that will easily to monitor like which step that we had the problem and how to manage our analysis, right? So in here we have two questions. So the first here, Mr. Wahyudin. Hi, Mr. Wahyudin, are you there?
Hello. Yeah.
Hi, it's clear.
Yeah.
Okay, Mr. Wahyudin. So please.
Okay. The question to Mas Fahmi is good presentation. So I just wondering how is the accuracy of the evaluation, the result of the talking. So do you have any data, especially if you have the data correlating with the maybe in vitro with your
Yes.
next?
Okay, thank you so much for your question. Yeah, this is the in silico steps, and this is, I think it's very trivial one. So yeah, for my pipeline is filtering from the compounds that we have in the library to get some specific compounds that maybe have some potential for the next step. For example, for in vitro or etc. Yeah, from this pipeline we cannot get the insight of the validity of the drugs, but we can filter out from the so many compounds to specific compounds that may be potential for the next step. So we can save time and money in the in vitro process.
Okay, so it is just a speed up the process of the docking process, right?
Yes. Right. So we just want to speed up the docking, and we can utilize so many compound as inputs. So in this case, we have like 9,000 compounds as input.
Okay, so does it mean also that the largest the data that we have, the strongest or the more beneficial from this method, Mas Fahmi?
Pardon? Can you?
I mean that the largest the data that we want to evaluate,
Yes.
the more strongest this next flow should be, right?
The largest data that we can evaluate, the more
Yeah.
comprehensive that we get the results.
Okay.
So we can evaluate so many compounds, so we get more comprehensive results. So mostly I think in Indonesia we just specify the joking for someone for example, plants. For example, we filter like compounds derived from tomatoes or papaya one by one, and in this step we can filter like many compounds from so many plants in Indonesia in one. It's just one process.
Okay. Thank you, Mas Fahmi.
You're welcome.
Thank you so much, Mr. Wahyudin and Dr. Fahmi. So now we are moving for the next question. This is from Ms. Warda Savitri Poland. So this is about the Lipinski rule of 5. She said that I was wondering, how do other rules like GOES or MOOC fit into drug likeness evaluation? Are they also widely used, or is it only Lipinski rule of 5 that generally considered sufficient?
Yeah, I think this is part of different rules. Like I think in GOES and we give it filters, it has different or more criteria than the Lipinski, and we can also use that for the evaluation. It's just like in the step of the process that we do. And in this step, I use the Lipinski because it's the common one, and I also can include another fitness to my pipeline if I want. Yeah. I think this is a good information for me, and I can fit also optional filtering process. Like maybe I can use GOES or another process that I can include in my pipeline.
Okay. Thank you so much, Dr. Fahmi, and also Miss Warda. So it's basically like our personalized manager, right? So we can
Yes.
customize our analysis.
Yes.
Thank you so much. So about the next flow or anagenic, is there any question from anyone? Okay, if there is no question anymore, so thank you so much, Dr. Fahmi, for your great talk and share. It's really happy to have you here. Thank you so much.
Thank you.
Okay, everyone, as we come to the end of the today's webinar, we would like to extend our gratitude to all of our speakers for their outstanding presentation and also valuable time. And we also acknowledge for all participants that already active for engagement throughout the day. But before we part, we'd like to invite everyone to turn on your camera for a group photo. I will give it time. So Please, everyone, could you turn on your camera so we can have a photo session here? Okay, I will wait for the 10 seconds. All right. Okay. So for anyone that hasn't opened the camera, let's go. Please open the camera. Okay. Okay. Okay, I will count. We will take a 2 photo, okay? So I will count, start from 3, 2, 1. Thank you for another shot. In 3, 2, 1. Thank you so much, everyone. And I also mentioned like previously about the door prize. Please don't forget to share our event in the social media, and also don't forget to tag our account so our team will decide who will be the winner. And finally, thank you so much for everyone. We are looking forward for the next event, and have a good day, everyone. Bye-bye. Thank you so much.
Bye-bye. Thank you.
Thank you.
Thank you, everyone. Have a wonderful night.
Bye. Thank you.
Bye. Thank you so
Bye.
much.
Thank you.
Thank you.